Good morning, distinguished guests, ladies and gentlemen. Welcome to the Open Space 2021. I'm Dr. Tony Ho from the College of the Science and Technology Hong Kong U Space, and I will be the MC of this keynote talk this morning. May I have your attention that could you turn your cell phone to the silent mode for the in-venue audience? Thank you. It is our great honor and pleasure to have Professor Danny Chan, Assistant Dean, Research Postgraduate Studies, Lee Ka-shing Faculty of Medicine, the University of Hong Kong, S.Y. and H.Y. Chan Professor in Stem Cell Biology and Regenerative Medicine at the School of Biomedical Sciences of the University of Hong Kong, to give us an inspiring keynote talk of stories about DNA. After graduation from the University of Melbourne with a Bachelor of Science, Master of Science, and PhD, Professor Chan continued research at his alma mater on heritable skeletal disorders with a focus on extracellular matrix proteins. He joined the University of Hong Kong in 1998, maintaining his research in skeletal biology using mouse model to address disease mechanisms in vivo, as well as human genetic studies to define genetic risk factors for common degenerative skeletal conditions such as intervertebral disc degeneration. In recognition, he was presented with the Premier Award for Health and Medical Research in Australia, and more recently, he received his Croucher Senior Fellowship from the Croucher Foundation as recognition of his contribution to skeletal research in Hong Kong. Without further ado, shall we welcome Professor Danny Chan, please? Thank you.
Well, first of all, thank you very much, Tony, for that nice introduction. And certainly it's my honor and delight to give this opening presentation for the open day of the Hong Kong U Space. I'm going to use English, my apologies, because 我哋中文都係麻麻地嘅咋. So I think, if I try to do this in Chinese, it might actually hard to follow me. Okay. So, as Tony said, I'm going to talk about stories about DNA. Now, there are many, many stories about DNA, and what I wanted to do today is give you a little bit on history, and then pick out a few things that I think is interesting for you to not only to think about it, but see what you can learn from it, and perhaps you can do something about it. Okay? All right. So I'm a professor at the School of Biomedical Science. I'm also the current director, so I look after the school, but at the same time, I still do have a very active research lab. And this is also part of my interest. And I have an active teaching program as well too at Hong Kong U. And also I do some teaching at Hong Kong U Space. I think this is very important, a part of the continual education as a lifetime learning process. So it's extremely important. And I'm really, really pleased to see some audience today that I can talk to. And I know there's quite a number of people on Zoom as well too. So thank you very much for attending, because it's nice to talk to a live audience. Yeah. Okay. So I am a developmental biologist or developmental genetic biologist, interested in looking at the skeleton, particularly rare and common disease of the skeleton. Okay. Now, in the picture that you're showing here, many you can relate to that. Okay, you're working with genes, and you're working with the skeleton, you're working with rare disease, and you're working with population genetics. Okay. There's also a little picture there I quite like. It's a little worm. It has no skeleton, but also very much interested in researching in this. And this little worm, I learned a lot about stem cells, because this worm has an enormous potential to regenerate. You chop off its head, you grow a new head. You split it in half, it duplicates the other half. It's really wonderful biology. And why would I be interested in that? It's because I'm interested in extracellular matrix, as Tony said. And extracellular matrix provides the environment for the stem cell to live in. And this little animal, as an adult animal, contains 30% of its cells as stem cells. So it's really lovely to work with it. And we just recently published a paper in PNAS. But this is as much I'm going to tell you about my research today, all right? Because otherwise you'll get bored. But I will tell you a little bit about the vocabulary that I use in my research, and some of these sort of terms and words that you may want to think about, and sometimes you may want to remember that, because I think it will be useful for you to know some of these terms. Genetics, genomics, genome, epigenome, epigenetics, rare disease, and so forth, right? Of course, what we want is really therapy at the end of it. For me, working with the skeleton, with all the diseases associated with skeleton, whether it be rare disease, whether common osteoarthritis, osteoporosis, intervertebral degeneration. As we get old, we suffer from this, right? And I'd like to come up with some sort of solution to this. But many of the things that we do is actually in that middle circle here. It's a process that happens within the cell. And that process involves the conversion of the information that encoded in our genome, the DNA. The DNA is then converted to RNA. And I hope maybe much of you learn about messenger RNA more frequently these days because of the vaccine, right? Okay, a messenger RNA vaccine. And the messenger RNA vaccine can be converted into proteins, okay? And protein are the working horses of the cell. And this is what I do within the lab. So that is a very, very important process. So that process takes place in basically all living things. Okay, the conversion of DNA to RNA to proteins. Some may skip the DNA part, like the viruses. Okay, so the humble viruses, or not so humble viruses sometimes, right? Okay, some of the viruses live within our genome, and some live outside our genome. But they have their purposes in life. And there's also the bacteria. They could be pathogenic bacteria, or they could be the good bacteria we call probiotic and so forth, right? To the vegetations, the plants and so forth that basically utilizes sun energy to convert the carbon dioxide that we breathe out and convert it back to oxygen. This is very, very important. I think the fish, what's it called? Nemo, whatever. And our best friend, the dog, canine. And of course, our descendant. Now, this process happens in all of these cell types. So this really in all living organisms. That conversion from the genetic code to messenger RNA to protein. Very, very important process. Now, living things are made out of cells. And these are some pictures about cells. And whether it be a nerve cell, whether it be a bone cell, whether it be, plant cell, cancer cell, whatever. This same process would take place. So cells come together to form tissues. Tissues come together, form an organ. The organ come together, different organs, to give a living organism, right? Okay. So that is that sort of process. Right? And in human, there are many cells enough. So there are around about 50 to 100 trillion cells. You can imagine the number of cells that are needed for us to become human, from a single cell to 50 to 100 trillion cells before it becomes us. Okay. So what's inside a cell? So this is a very simplistic picture of a cell. So inside a cell, there is the nucleus. And the nucleus is where our genes are. That's where the DNA exists, okay? Right. So inside the nucleus, something will take place that converts it to messenger RNA. It's called transcription, that process. It's inside the nucleus. And the messenger RNA then get outside the nucleus, and then it gets translated into proteins. So there is, I can't read it, there is a organelles called the Endoplasmic Reticulum. And that's where most proteins are made. Yeah? And there's also a organelle called Golgi apparatus. And that Golgi apparatus helps protein to be secreted. If protein that need to be functioning outside the cell, it need to be secreted, all right? Okay. And there's other organelles that does things. But inside that cell, there's that cytoplasm. That's where all the goodies are. Okay? That's where the proteins, the metabolites, the energies, and so forth. But the energy is made by another organelle called mitochondria. And I highlighted there. It's in red. It's very important. This is the powerhouse of the cell. These organelles produce most of the energy needed for the function of the cell. And there's also a story about where these mitochondria come from. And if you look at these mitochondria, you say, it looks like a bacteria, right? Okay. So there is this concept, the mitochondria, in fact, was originally a bacteria that works symbiotically with the cell, and after a while, it becomes the part of the cell, okay, for the functionality of the cell. So, but the mitochondria and the nucleus are highlighted in red, and the reason is that there's DNA in both of these organelles. While most of our genetic materials is in the nucleus, the mitochondria also contains an additional set of DNA. And I'm going to tell you a story about the mitochondria a little bit later. So we know that there's about 21,000 genes that they all sort of pack inside this nucleus. And every cell has a complete genome. Every cell has a complete genome. And that genome is exactly the same for every cell. Okay? So this genome contains information because it contains information about us, and they are represented by basically sequences of DNA, and they're represented by 4 nucleotides, ATGC, right? Okay, and they pair up in a very specific way. But basically, the different combination represents, what that DNA sequence can function. And it normally functions as a gene. And a gene have a defined position within that sequence. And here, I've got here a start and a stop. Okay, that start and stop really part of the gene that is coded for the protein. That's the start of the protein and end of the protein. Start of translation and stop of translation. So that is the basic sequence of DNA. And the DNA is then wound into a double helix, and then also shown here that it also twines within these sort of little barrel-like protein structures called histones, okay? And the DNA wraps around that histone into little balls like that you see in that slide. And then it certainly supercoils again into this old telephone core that you see, and then it's assembled into a little cross structure at the top that looks like a chromosome. Okay, so DNA is the language of life, and is organized into chromosome packed into the nucleus. So this is the human genome. There's 23 pairs of the chromosome, and the mitochondrial DNA as well too. Okay, so there are the 21 what we call chromosomes, and there are 1 pair of 6 chromosomes, the X and the Y chromosomes. Okay, for ladies you have 2 X. For men, there's an X and Y. All right. Okay, and that defines the difference. But chromosomes, DNA doesn't always organize in chromosomes. Chromosomes is only organized in a particular phase of cell division, in particular during the metaphase, when they do pair up together, ready to be pulled apart to form 2 cells and so forth. But normally, this is what it looks like, okay? DNA are packed in the nucleus like this, like a bowl of noodles, okay? But if you look at that bowl of noodles to the right, that is different color. Okay, so even though they look like a ball and they have positional space. So the different color represent all the DNAs within a particular chromosome. So they do have physical space, what we call 3D DNA structures. And within this, they also have what they call the, I can't read it. But anyway, the epigenome space. So you could, so what they call where the DNA is active or inactive. So you could sort of, because not all part of the genome is active. Okay, I will explain to you why. And then you can unfold it a little bit more to the chromatins and then the sequence structures, right? So, and how the gene is expressed controls what cells they are, right? Because we have different types of cells, and yet they have the same genome. So how could they be different types of cells? And the reason they are different types of cells because different sets of genes are turned on and different sets of genes are turned off, okay? And therefore they are the cells that they are. Okay. So this is a diagram representing a stem cell that can become many, many different cell types, all right? So for example, the totipotent stem cell that when a sperm is an oocyte, gets fertilized, and that one cell then becomes all the 50 to 100, trillion cells of you, whatever cell that may be, fat cell, bone cell, blood cell, neural cells, whatever, right? How is that possible? Now to explain that, I think this is a very nice diagram that most cell biologists use, is the Conrad Wellington epigenetic landscape. So one can imagine that, a stem cell, a multipotent cell, sitting at the top of a hill. And that hill has many, many valleys and bumps on it, right? Okay. And at the bottom, there's lots of space. So basically, if that cell at the top rolls down that hill, it depends the path of the valley it takes, where it ends up at the bottom of the hill determines what they are. Okay, so you can see that a hill, a green cell can become a red cell down the bottom, or becomes a purple cell down the bottom. But you can take different pathways of getting there, right? So every pathway it takes, and every turn that it takes, different genes comes on and off. And certain gene will maintain on to keep it that cell. Okay, so the lineage and path of stem cells depends on what genes are turned on and what genes are turned or maintained on to keep the cells as they are. All right, that's enough background. What I want to say a little bit now, tell you a little bit story about DNA. There's so many stories about DNA that I want to just pick a few, but I want to just touch a little bit on the history of DNA. And these are just some of the people which I think play a very important role. Okay, of course there are many, many. I'll take you back all the way to Charles Darwin, okay, and so forth. And then tell you a little bit about decoding the genome and what's fun about decoding the genome and what have we learned. So I begin with what I call the pioneers. Okay, the pioneers includes, I felt it would be Charles Darwin, who gave us, that really fundamental concept of the origin of species, evolution by adaptation, fittest survival, so forth. And then there's also the work by Gregory Mendel, a monk who's very interested, is a geneticist, very interested in looking at peas, shapes of peas, colors of peas, and they work out the basic principles of genetic and hereditary. Okay, he worked out the principle for dominant inheritance and recessive inheritance. And then there is Frederick Mitchell. He did a very, very important thing because he identified DNA. He called it nuclein then, right? But he showed that DNA could be the hereditary material to explain genetics. And there's also Sir Archibald Edward Garrott. He was the first one to use human diseases to illustrate Gregory Mendel's principle by studying a disease called alkatonuria. It's a genetic disease, it's a metabolic disease where there's a problem with the enzyme that's not able to do some conversion of to urea. So your urine becomes dark when it gets exposed to oxygen. Okay, and these people actually have some form of osteoarthritis. So I'm actually quite interested in that but it's a metabolic process. But he was the first one. But of course there are others too. Okay, whereas Oswald Avery determined that the DNA is a transforming material that can be, convert a bacteria, a pseudomonas from a harmless to a pathogenic strain just by changing its DNA. Very important. And lastly, Edwin Chargaff, where he really defines that DNA composition can be different between species, but it has a rule. That rule is that the combination of A, the amount of A's is also equal to T, and the amount of C is always equal to G. That's the pairing. He didn't know it then, but this is a very basic, fundamental information that allows what I call the next discoverers to really come to terms with the structure of DNA. We know when we talk about DNA story, we always talk about, the structure of DNA, Watson and Crick structure and so forth, right? But their discovery is really based a lot on that fundamental information that we got from the pioneers. Okay? But in addition, a few people that we kind of ignore is really the work of Rosalind Franklin, okay, where she able to crystallize DNA, and because you were able to crystallize DNA, you can use X-ray diffraction pattern, and from the pattern you can get some information about how atoms are organized within a protein or DNA in this case here. Right? So I think we should really give credit to a lot of people who really enables James Watson and Francis Crick to define the structures of DNA. Okay, so, and also the fact that Marshall Nirenberg, he figured it out, what the coding of the DNA that can be converted into amino acids. So that's very nice. So that's a picture from the picture that he drew to really now the structure of DNA, which is the double helix. And this is the same structure in all cell and almost all organism. This is the major structure, but it's not the only structure. There are other different structure for DNA as well too. So why is that so important? Because understanding the structure of DNA, now we can now begin to, this is sort of pairing, and that the sort of opposite strands of DNA tell us that, hey, one is the mirror of the other. It's a copy, but it's a mirror of the other. So that means that if we can begin to use that concept to really begin to understand how DNA could be replicated. So if you can break those two strands apart, and those become a template in which we can copy it, right? Yeah. And this is exactly what's happening. So we can, from the structure of DNA, now we'll begin to understand how DNA may be replicated. That is applicable to cell division and so forth. And that's life. And by understanding this, we can also now understand the concept of genetic variation and talk a little bit about that, evolution as well as hereditary. So when DNA replicates, DNA needs to split apart. So basically you break it open, and then as you open it, you start copying the other, right? So basically the one strand of the, one copy becomes two copy. And that's very nice, and that's part of it. And we do this for the 3 by 10 to the 9, just, number of bases in the human genome. There's a lot of copying to do, right? A lot of copying to do. So a lot of copying to do means that there's room for error. And you do make error. Although in the cell, there's a lot of what we call, mechanism to ensure accuracy, right? But errors do take place, and they introduce what we call polymorphism into the DNA, variations. It could be just a single nucleotide variation, we call SNPs, or it could be deletion, insertion, and what have you, all right? Okay. So at the moment, we know that there's lots of them. There's about 4 by 10 to the 7, even just single nucleotide variation between you and I. So there's lots of them. And then comes the technologists. Now the technologists, the basic, to me, the most important technology was DNA sequencing by Frederick Mitchell. He devised the most clever way of sequencing, the dideoxy sequencing. There are other sequencing methodology, but I think this is really one of the nicest. When I used it when I was doing my PhD, it's just so wonderful to be able to sequence DNA at that time and to read it and get so excited every day. So a new sequence every day. It's just so exciting at that time. Really, really nice. Okay. And then because of the fact that we can sequence DNA, and at the turn of 1900, someone said, well, why don't we sequence the entire human genome? Wouldn't that be nice? Okay, so there was lots of idea going around, and then in the end, it was decided we should do it, and you should do it at a public consortium. So the Congress and the Senate come together to put some money, say, look, you guys should do it. Okay, and it started with James Watson, but then it was took over by Francis Collins, who was the director of NIH at that time. So they decided we should do a public sequencing of the human genome. And they calculated from the rate that they can sequence, they would take them 15 years. 15 years, okay? And they calculated they need lots of money, so they convinced Congress and Senate to be able to do this, get enough money to do it. But 15 years is a long time. But it was a very systematic, very scientific, involving numerous countries, 6 countries, and lots of labs. You do a bit of that, you do a bit of that. And at the end of the day, we'll put together, right? We're very systematic. But around about towards the late, just on the turn of the millennium, come Craig Venter, an entrepreneur, and say, you guys are doing it too slow. Too slow. I think there's a better way of doing it. With computation ways, so why do we need to do it systematically? We'll just do it randomly. We'll call it shotgun sequencing. I say, we'll just do shotgun sequencing. We'll ask the computer to put it together. He's an entrepreneur, so he's exactly that. So he's got now private money to do it. Okay? So you guys will take 15 years. I will just take 1 or 2 years to do it. Yeah? So he put a bit of pressure on the public side. So in the end, I think both really by 2000, both end up with the first draft of the human genome. And by 2003, we have the first complete draft of the human genome. Okay, which really sort of match with the 50-year celebration of the discovery of the human genome. So this is very nice. But what have we learned? $3 billion that costs. That's a lot of money. Okay, what have we learned? Okay. So we've learned that we have a book of DNA, and everyone can have a book of DNA if you wish to, if you want to pay for it. Okay. And because of the fact that we can do it, and because the technology is increasing and so forth, there was a concept that why don't we actually sequence more? Okay, it's the 1,000 human project was initiated in 2007, and it was completed in 2012. So from that, we've learned a lot. So there was a just over 1,000 human genome project, and we learned that there is really a lot of information about it. But we also learned there's also a lot of differences between humans. Okay, and that differences give us each one of a genetic signature, and that genetic signature of us, how we differ from people from the rest of the world. And that's very important because that represents you, right? And because of that, we learn a lot of things. So we learned that, in fact, amongst all the different people here, different ethnicity and so forth, we are similar to about 99.5%. There's a 0.5% difference between every one of us. Okay. So these differences actually very important because that's tell us that we're different, tell us different one and apart. And it tells us also our susceptibility to disease. Okay? Whether you're more susceptible to diabetes, depression, cardiovascular disease, osteoarthritis, osteoporosis, and so forth. Okay? But some of us, unfortunately, have what we call rare conditions. Okay? These are very drastic conditions, but you can also modify the conditions as well too. Okay? And some of us have private variants. Only I have it, nobody else. All right. Okay. So this is very important because this variation actually tells us a lot of things. So this picture I got here tells us that, hey, we do look different, right? So you can hear, if you look at the faces of this lady here, you could say, well, the lady on the left, Caucasian, the lady in the middle, maybe in the far right, second one may be Asian, and the darker color one African. So you can tell already, right? Okay. But if you look very carefully, it's the same face. It is the same face. It's only different in color. Okay. So we can tell the ethnicity. So we have very specific characteristics that tells ethnicity. So I'm a scale of people. Right? So here, in addition to color, hair, and so forth, your skeleton, the shape of your skeleton, they form during development. It's also dictated by your genetics and also dictates ethnicity. So that you could look at person, even Asians, I can tell you, hey, you're Japanese, you are Korean, you're Cantonese, you are Northerner, right? There is that certain characteristics, all right? Just from the face itself here. So the craniofacial, we call it development, from the structure and shape of the bone together with the color of the eye, hair, and so forth, really defines a really classic features of that, okay? And you know what? You can actually predict your feature, your facial feature from your genome. All right. So that can also affect health and your response, whether you can, feel pain more, or whether and so forth. So there's lots and lots of things. So from this variation here, we could link genetic variation to what we call phenotypic traits. What do you do? How do you feel? What do you look like? All these things. So we have all this information amongst all the thousands of genomes that we have. We could begin to make association between a variation to a particular phenotypic outcome, right? Okay, and this is exactly what we wanted to do. And this is what genetic tests do. Okay, so genetic tests tell us the variation that you have and what you're likely to have. I don't know whether many of you like to eat asparagus. Okay, and tell you, for those who eat asparagus, if you go into the bathroom and you pee, some of us can immediately smell it. I have eaten asparagus. Yeah. I don't know whether any of you experienced that. Okay, it's very strong. Some of us can't. Some of us can. And that's the variation that you have in your gene that tells that whether you can or you cannot. Okay? Or any one of you actually suffers when you get up in the morning, pull the blind and get, okay? That snip, if you have that snip, you have the tendency to do this. Okay? That change in the light makes you sneeze. Okay? Or you have very dry earwax. Or there are other snips that say, I like broccoli and I like Brussels sprout and I hate broccoli and I hate Brussels sprout. Okay? All right? Or I can see green and blue and you can't see green and blue. These are all variations. These are fun variations. And this is really the basic of a lot of genetic tests that are around. I don't know, you get ads and say, look, you want your genes tested. And you can get your gene tested. And there are many companies that offers our services in Hong Kong, in mainland China, in US, and so forth. You could spit into a tube, okay, send it away, and you get some information back and say, look, this is what you have. You got good muscles, and you have likelihood to have type 2 diabetes, family tree, or you can do your hereditary, whether you have partly this, partly that, whether you have any European descent, you have any sort of other sort of descent, so forth. Okay, and these are fun. Okay, these are information, and you've got to be also very careful how you interpret such data as well too. Okay, because they are not as accurate as you think they are. In particular, your ancestry things, and ancestry really depends on recombination. Because sometimes, just say you're 30%, or you got no percent. So I'm sure my parents from one of my parents from Vietnam, but how come I don't have any Vietnamese genome in me? You get a bit worried, right? Okay, but these are not, because it really depends on recombination and so forth. Okay, so this is nice, but it's I think what we wanted to do is a link to genes and diseases, right? So those things are what we call common and chronic diseases, and diabetes, hypertension, arthritis, and what we call common, chronic, and complex, because not only one gene does it, a combination of genes coming together with the environment. But I work with rare diseases, cystic fibrosis, Marfan, Huntington, osteogenesis imperfecta. These are rare diseases. These are quite not so nice diseases to have, because you have it, for a long time. And many of the technologies that we have these days, DNA technologies and healthcare, it's really driven by the need for healthcare. I think I have time. Let me just tell you a little bit. I think this is fun. So we have the live genes. The genes actually does things, but you probably don't know that there's lots of dead genes in our genome. Okay, we call pseudogenes. So they're called basically molecular fossils that are die and they are still buried within our genome. And we have many dead genes in our genome as live genes, basically. Okay, and one of the really interesting dead genes is a gene that is needed to make ascorbic acid, vitamin C. Now, we know that you say, you got to get your vitamin C because you can only get vitamin C by eating fruits and vegetables, right? Okay. Long time ago, our ancestors were able to make vitamin C, but we lost it. We lost it about 60 million years ago. That little arrow there, that split, okay, between us and the lemur that were able to say, still able to make vitamin C. So now we are completely dependent on that. Why we have lost vitamin C, don't know, because it's based on Darwin's theory, it's survival of the fittest, right? But there are also mutations. There's mutations always there, right? But for some reason, this mutation, the inactivation of an enzyme that needed to make vitamin C was selected for. I don't think there's any selection in terms of advantage, because there's no advantage on that. Okay. But I guess at that time, the earth is very warm and the tropical forests are expanding. There's plenty of fruits and vegetables. So our ancestors, the primates, really don't need to make ascorbic acid. If you don't need to make something, why doing it, right? It's a waste of energy. It's just like if you don't use a muscle, you will lose a muscle because you don't need it. So let's not waste energy. So we lost it. So we are very much depending on our fruits and vegetables for vitamin C. The other dead gene is this gene, UOX. Now, I don't know anyone you have ever suffer from gout. Not nice. It's because we don't have this gene. So therefore, our concentration of uric acid in the urine or in the system is very, very high. Okay, and therefore, when it's high, when the temperature is a little bit cool, it forms crystals, crystallized cell, and it's really painful in joints. All right. Now, this gene was lost a lot later. So, GORA, which is a gene that makes ascorbic acid, we lost it about 60 million years ago. Okay, LOX, which is a gene called urease, we lost it about 17 million years ago. All right. Okay. It's lost against due to mutation. So basically, this gene slowly die. But it's thought that this gene had an advantage for us. Losing this gene has an advantage because as the Earth gets cooler and so moving further north, that having this gene allow you to be able to convert fructose, which is present in fruit, and convert fructose in fat and store it for times when you don't have access to fruits and vegetables. Okay, so it's an added advantage. It's also an added advantage that it's actually supposed to make you a little bit smarter. Okay, Don't know whether it's true, but if that is an advantage, that means that, from a genetic point of view, sometimes less is more. Losing that does give you advantage, but losing that also give you side effects, right? All right. So there was other genome that sequence. That's nice, and basically this forms the species tree evolution, and this provided the molecular outlook of Darwin's theories of evolution. So this is very, very nice. But if you take a little bit closer, our closest relative is the primates. Okay, so if you take the macaque, we have about 90% similarity to the macaque. But our closest relative is chimpanzee. We are 98.5%. Okay, right. I'm 99.5% similar to you. That one difference, that extra 1%. Okay, right. So we're not. The other thing is also very interesting. So this is me and my wife, male and female. Male and female is 98.5% different. But you tell me, okay, difference between people is 99.5%. It's true. It's 99.5% between males. All right? So that compares to me and Albert Einstein. Obviously, that 0.5% is huge difference, right? All right? Right, so it's huge, huge, huge difference. But you say, well, me and the chim is 98.5%, and me and my wife is 98%, all right? Okay? Hey, this is something that I can use when I have a fight with my wife. But it works both ways, all right? Okay. It's just a bit of fun. All right. Now, having differences is also very, very interesting because now we can begin to understand a little bit about evolution of people. And Brian Sight is a friend. Unfortunately, he passed away a few years ago. Brian Sight wrote this book called The Seven Daughters of Eve. Now, he's a geneticist. He's also interested in the skeleton. Okay, and so we meet at conferences. So here's this idea, he's looking at the mitochondria. Remember the mitochondria? I said the mitochondria is something very interesting. The mitochondria only comes from your mother. Yeah? Okay? It does not come from your father. So your mitochondrial DNA is always come from your mother. It's a paternal thing. Right? So this book is half non-fictional, true, and half fiction. But what he used is that he used study mitochondria from different people in Europe. And he come up the idea, and in fact, everybody in Europe, Europeans, come from 7 women. Okay? Basically, he's looking at the genetic, the haplotype, and say, look, you guys coming from 7. So these are the 7 daughters of Eve, the mitochondrial Eve. And this mitochondrial Eve lives in somewhere in Africa. Yeah? And thus supports the so-called out of Africa hypothesis. Okay? And there's also the so-called Y chromosome Adam, because the Y chromosome comes from the father only. So by combination with the mitochondrial Eve and the Y chromosome Adam, it actually fits the so-called outer Africa. So this shows you how human migrations out of Africa, and the number represents the year, and the size represents how many people you populate. So, some interesting information there. So changing in the genetic programs to tell us lots of information, but is it good or is it bad? So obviously I tell you this, it can be good, it can be bad as well too, all right? Okay. But the question is, has human genome and evolution, have we evolved? Have we evolved? Are we evolving? Okay, and have we evolved recently? And I don't think we have, okay? Basically because I think this is a concept by Sidney Brenner, which I like a lot. And he just says that we are too smart for our own good. Why? Because basically we are now in a era of what we call maladaptation, the environment and disease. Okay? And that we are living too long. Okay. The issue is that now when people start to think, when we're able to alter our environment, we have stopped evolving. Because we evolve because we adapt to the environment, okay? But now we're changing it. If I feel hot, I will slowly adapt. But no, I turn the aircon, okay? All right, okay? If I feel cold, hey, I should grow hair to protect it. No, I kill an animal and wear the coat, the fur. Right? So basically now we are, adapting ourselves to the environment. We are not evolving, okay? And we're living too long, okay? We're not supposed to live that long. Basically, we're animals. We're past our reproductive age. We're no good anymore, right? But we're still around. So basically, all the chronic disease because we are really live longer than we should. Okay. So what can we do about adaptation? We can fix the gene, for sure. We can change the genome to fit the environment. But is that the solution, fixing the genome to change it? Or should we use the environment to fix the gene? If I have 5 more minutes, I want to actually tell you something very interesting about that, okay? We'll patch up the phenotype. And this is exactly what we're doing. Modern medicine is patching up the phenotype. We're keeping a set of weak genes in the pool. We're not evolving, unfortunately. All right, okay. So what can we what control? So there's a concept that, gene does everything, or, that you get your gene from your parent. But I think we need to begin thinking a bit different between nature and nurture. Okay, and I want to tell you a little bit about the importance of being nurture, right? Because gene is not your destiny, at least not the rare disease. If you have a rare disease, that's very hard. But most of the common thing is not your destiny. Okay, and it's called epigenetics. Anyone of you heard about epigenetics? Good, all right. Epigenetic means it's above the genome, meaning that we can actually alter information beyond the genome and something that we can control beyond the genome, all right. So the classic example is that, these are identical twins, but sometimes identical twins are different because their genes are controlled differently if they are in different environment, okay. And I want you to give you information that in terms of how epigenome works. Epigenome work is because there's the histones that the DNA wraps around, and the histones gets modified. There's modification in it such that it controls whether the DNA is open or closed, so the accessibility for that gene to be expressed. And the other is here, methylation. The DNA also gets methylated. Okay, in it, and that methylation also suppresses its expression. I want to say something about DNA methylation in this mouse. This mouse is really interesting mouse called a agouti mouse. So the agouti mouse has a gene, the agouti gene, right? And if you have this gene, the poor mouse is yellow, but it's also fat, very obese, and have a short life. Okay, and there's another mouse here. So the interesting story about this mouse, and I don't have too much time to go into it, but these two mice have the same genome, different mothers. And the different mothers are fed in different diets. So one is a normal diet that we give, and the other one then is yellow and fat. The other, they gave what they call methyl good supplements, and look, normal, thin, dark color as you should be, and have a normal lifespan. So what the mother eats change the outcome. What the mother eats change the outcome. And what the mother eats is the methyl groups. The methyl group, the methyl donors that talk about the methylation of the DNA, right? So if the mother eats a lot of these vegetables that has folic acid, and B vitamins and so forth, it alters the outcome of the offspring completely. Yeah? So what you eat is what you are, but also your mother. And I also put grandmother. I don't have time to go into that, but what your grandmother eat also influenced you, because your mother was inside your grandmother. Yeah? All right. So, in addition to what you eat, whether you do exercise, whether you're stressed, whether you're happy or sad, all influences how gene expresses. Okay? So you can do something about it. Have a laugh. Don't be sad. Do a bit of exercise. Alters your DNA destiny. All right. So basically, I think genetics and genomics have a huge role in healthcare. I don't need to go into that. I think most of you understand that. And this concept of precision medicine is here to stay. Okay? We will use a lot of information that we have from biobanking, understand more about genetics, understand more about the role of DNA sequences, the information in there, and because next generation sequences is really cheap these days. And with the power of computing, we can really understand it. We can edit the gene, whether we should or not. It's possible. There's pharmacogenetics, meaning that we now, if we know our genes, we know exactly what sort of drugs that we should take. Maybe a bit of folate, a bit of thing could help, right? So by understanding that would really help. So meaning that health profession, not only health professionals should need to understand and be aware of genetics, we as a community should do as well too. Okay, this is my last slide, the future. Is managing your genome. So it's like playing cards or have a game of mahjong. Okay, what you begin with, what you deal with is not the destiny. All right, okay. You know the hand that you get in mahjong. It really depends how you play the game, whether it be a loser or winner. So you understand how to win, you have a better chance to win. So healthy life, healthy aging. That's the last of my presentation. So thank you very much for listening.
Thank you very much, Professor Chan, for your phenomenal talk. I'm sure all the audience have been impressed by the fascinating stories from the book of DNA after this keynote talk. Now we will have Q&A section. Okay, so I think any question from the audience in venue?
So Professor Chan, thank you very much for that fascinating story. So one thing, when you talk about people can smell asparagus, so one thing came to my mind. So for people who like durian and for people who cannot stand it, is that because of genes?
I think people like is always very much in genes because it's what you get used to, right? Because, you know, I like certain smells and I don't like certain smells. Durian is one thing, okay? You know, it really is, it's something that you're born with, but something that you can change as well too. Okay. I usually don't like Brussels sprouts as a child, but now I love Brussels sprouts.
Because your gene has changed or because of?
The modification has changed because this is all due to modification, epigenetics.
Is that intended or just by accident?
It's exposure, right? Because it's called what we call adaptation. You adapt to that. Yeah. So epigenetic is something that you can change, and it's also reversible.
Okay. I don't think I'm going to adapt durian for the rest of
If
my
you
life.
eat it long enough, you will.
No. I'll never try it. Okay. The other thing is just some thought. Whenever I don't like something about myself, I will put the blame on my parent. And then they will always say, sorry, you don't have any choice. So I'm thinking, after listening to the story, I'm thinking, in the future, do I have a choice?
I think there is always a choice, right? So this is really this last slide here is what I try to illustrate. So what you dealt with, whether it's a deck of cards or the 13 pieces that you get in mahjong, is what your parents give you. That's it. That's what you got to work with, right? Okay. But you can change that to some extent, over time. And I think this is what maybe you can learn by understanding your genome. Yeah, your genome tells you that you're more susceptible to this and so forth. And if you could just change your lifestyle a little, you could help to prevent that. Yeah, it may not necessarily resolve it. Yeah, whatever you might say are more susceptible to a particular chronic disease, you may not able to change it completely, but you could say, I can delay it, I can decrease its severity. We all get old. Nothing we can do about it. That's something you cannot change. You will get old, we will die, okay? But it's the process along the way that we can do something about, right? Okay, delay a little, and then even when it starts, the process will start, it's less severe, okay? And by the time I die, I end up with a more healthier life as an age. Okay. Yeah. Unfortunately, we are not a healthier, as we go, although medicine definitely helping us to extend our life, but really we are not healthier. I don't think we as a population is a healthier population.
May I have a question? Yeah. Thank you, Professor Chen, for the very informative talk. I want to ask a question about the pseudogene. I fully understand that we lose the function of some gene due to either we got advantage or we try to save some energy, don't make unuseful stuff. But why we didn't lose that sequence? We completely lose the sequence or got to remove that gene.
Okay. I think it's maybe I could put this in an analogy of a hoarder, right? Hoarder, right? Many of us hoard things. We don't like to throw them out, just in case, kind of thing. So as with evolution, there's two ways. Either you delete it or you duplicate it, right? And in a lot of cases, it's the duplication that does things because you want to increase function rather than decrease function. Okay. So duplication is always there so that you've got multiple copies of the same gene. And then when you have duplication, it's also as a backup. If one fails, it still can working. But there are still many genes that are just one. Right? So I think not losing it is probably the way in which it sells as a backup system. I think actually reactivating some of the pseudogene may be beneficial. And one of the really classic experiments I did was the uric acid, the uricase that die. Because when the scientists did reverse genetics, meaning they go back and find their ancestral uricase, and they corrected this in the pseudogene and turn it back into a functional gene and put it in cell, and guess what? It does wonderful things. It works better, really quite active, right? So I don't know, in terms of say, we are not evolving, but can we force the evolution? Yeah.
Hi, so I have a question about the epigenetics. So the genetic code, our genes will be passed on from generation to generation. So, but I have a question will any of the epigenetic code be passed on to the next generation, or is it unique by person and it's entirely different?
Epigenetic can be passed on. Yeah.
Right.
So what, is that example of what you eat is what you are, but what you eat is also what your mother eats and what your grandmother eats. Yeah. So a good example is that your mother that generated you, her egg was generated in your grandmother. And how that egg was generated depends what she ate. Yeah. So that information is really, so what your grandmother eats influences the oocytes in her that generated your mother, that generated you, and what she eat then generated you. And if, your sister or whatever, she would have your mother's influences on her oocytes that pass on the next generation and next generation and next generation. Okay, so that's sometimes why you think, hey, why there's a skip in generation? I look more like my grandfather. Interesting thoughts. So epigenetic is very important. Epigenetic is controllable, reversible.
Okay.
Yeah.
And also, I have one more question about like the pseudogenes and like. So I have a question about how much percent of our genome is actively coding, and how many percent of it is actually non-coding or pseudogenes.
Okay. So it's estimated only about 10% of the genome are coding. Okay? And another 10% of the genome are pseudogenes. Yeah. Why we still don't know much about the rest of the genome, and some of us call it junk, but I don't think they're junk. Some are junk, right? It's just leftover things. But what we do know now that the rest of the genome contains other things. MicroRNA is very important that I'm not touch on. The other thing is how the gene is regulated. It's called the regulatory sequences. And this information is how other proteins that can bind on to the genome that regulates the expression of that gene. It's called regulatory sequences. And they are scattered everywhere. All right. So, no, I don't think the rest of the genome is junk, and I don't think the rest of the genome is useless. We just don't know what they're doing. It's time, maybe you're young, maybe you will be the next scientist who will discover what the rest of the 90% genome is all about. Yeah.
Hello.
Thank you. From the online audience, this is a question. You mentioned humans are not currently evolving, but are this population in Africa, whereby the sickle cell disease gives protection against malaria, is this an example of evolution? Thank you.
Well, definitely. I'm not saying that we're not evolving. We are evolving. We're probably not as evolving as quickly and that we should. I think malaria and sickle cell anemia is a classic example that you basically in that particular region you need sickles, you need that mutation in your hemoglobin to survive. Otherwise you won't. But there's a lot of side effects, right? There's a huge side effects. Yeah. So unless you have that gene, you would not be able to survive in that region. So it's adapting for sure in that particular region. Yeah. And the other example is, the people in India, the color of the skin, it is evolving. It's just probably not evolving as fast as you can see the northern Indians and the southern Indian. They're very different in color skin, but yet they're the same. Yeah. So it is evolving. I'm not saying it stopped evolving, but certainly the way that we're altering our environment. Sickle cell anemia and things like that, we're not altering things. But as soon as we begin to alter things, alter environment to adapt to our environment, rather than in a way that is different, right? Yeah, I'm sure that our finger will be longer the more we use the iPhone. A thumb will become much, much longer for selfies.
I have a question. Thank you for the speech. But my question is, I believe our genome does not change as we age. It stays the same our entire life, but it does degrade over time, does it not?
Oh, I think, our genome, I'm not so sure we degrade. I guess in a way it's degrading, it's changing, because as every time the cell divides, there's a mutation. Okay? Although the accuracy of mutation is the accuracy of replication is high, we're still getting about one mutation every 10 to the 9 base pair replication. So over the lifetime of human, it's estimated to be, we accumulate roughly about 50,000 mutations. Yeah, okay, that's quite a lot. So yes, as we get age, we get mutated. My skin gets spotty, and so forth. So I mean, these are all changes. These are changing. Yeah, but these are either at the level of the DNA sequence, because ultraviolet or whatever, my skin getting spotty, lots of freckles and so forth, or the epigenetics being changed. Yeah, because also the environment, the cells also change. So it's all combination of things as we get on, I'm afraid. I'm not so sure that answers your question.
Well,
yeah.
I guess my follow-up question would be: is there a way to slow down that aging?
I think there's a huge body of research, right, on anti-aging, and there's lots of colleagues of mine doing that at the Faculty of Medicine, and there are other people around the world doing this by studying people with premature aging. All these things. There are genes associated with aging, and we know that. But many of the genes is actually influenced by diet. Okay, calorie restriction, for example, is one way. Not intermittent fasting, but calorie restriction, at least in animals, in C. elegans, in mice, do extend the life. Now, whether they extend the quality of life, that's another matter, right? So we like to live longer, but we like to live longer healthier. And basically, this is all the concept of healthy aging. We like, okay. You like to move, when you get old, you like to be mobile, and that's why I think the skeleton is very important. As you get old, you don't want to be in a wheelchair. Yes, you've got a good brain, but you sit in a wheelchair, it's not very nice. Yeah, you need to be mobile and so forth. All those things are actually quite important to me.
Oh, thank you, Professor Chan. I think we have been overrun, so
sorry about that.
No problem. So that's the end of today's keynote talk, and we will have a souvenir presentation. Thank you very much.
Thank you very much for listening and coming.
So may I take this opportunity to introduce our head of College of Life Sciences and Technology, Dr. Bruce Jung, to present the souvenir to Professor Danny Chan. Bruce. Thank you, Professor Chan, and thank you, Dr. Jung. Thank you. So that's the end of this talk, and hope you enjoy the rest of our open day events in the afternoon. Thank you very much.